Hello everyone, my name is Yaser Rawi and I'm presenting on chemotherapy-induced peripheral neuropathy. Chemotherapy-induced peripheral neuropathy, or CIPN, occurs in about 60 to 70% of cancer treatment cases, making it crucial to understand the molecular pathway to help prevent neurodegeneration. After about 6 months, CIPN drops to 30 to 40% of cancer treatment cases. Peripheral neuropathy is a significant dose-limiting side effect of chemotherapy, which means that the chemotherapy can't be utilized to the full extent and the treatment efficiency is reduced. Peripheral neuropathy is characterized by pain and numbness in the extremities like hands and feet. Many different classes of chemotherapy are very effective against rapidly dividing or cancerous cells, but also include the side effect of nerve damage, and it's not well known why neurons are negatively affected. Our goal is to better understand the pathway of CIPN through inhibition of certain proteins. Wallerian degeneration is a natural process in the nervous system that occurs when a traumatic injury to the peripheral nerves occur, but has also been recently found to occur in non-injury disorders. Nerve bundles are composed of axons surrounded by Schwann cells to create the myelin sheath, and fibroblasts make up the connective tissue. In order to regenerate the nerve connection after an injury, degeneration and removal of the distal axons and myelin sheath occurs, even though a patient didn't sustain an injury in the area. This degeneration is vital for the next step of the regeneration of the nerve bundle. SARM1's role in Wallerian degeneration isn't explicitly known, but it's understood that it plays a key role in downstream activation of axonal degeneration. So, on the figure on the right, an injury would occur to a nerve bundle, and SARM1 is activated. This leads to NAD depletion and a decrease in ATP, which then eventually leads to axonal degeneration. CIPN is likely to share many of the same mechanisms found in Wallerian degeneration. We have previously found that neurotoxicity as a result of cisplatin, another commonly used chemotherapy drug, requires SARM1 activation. Cisplatin also causes CIPN. The focus of this project is to try to find upstream regulators of SARM1 in hopes of preventing CIPN. So on the figure on the right, chemotherapy is the stimulus that triggers the MAPK signaling pathway. One kinase phosphorylates the next, eventually resulting in activation of SARM1 and eventual apoptosis of neuronal cells and the degeneration of peripheral nerves. So why is this important? CIPN is a painful and disabling condition with no known prevention or cure. An estimated 1.8 million Americans are diagnosed with cancer every year, and as a result, CIPN affects a majority of these patients. CIPN also limits the dosage of chemotherapy, which means that cancer can't be effectively treated without this condition. Understanding the pathway of CIPN has the potential to help with other peripheral neuropathies that result from diabetes, alcohol use, ALS, and other genetically inherited neuropathies. The main protein kinases we are interested in analyzing are HGK, or MAP4K4, MINC, or MAP4K6, and TINIC, or MAP4K7. All these kinases are upstream regulators in the MAPK pathway, leading to the stress-induced CJUN N-terminal, or JNK signaling pathway, which leads to SARM1 activation and eventually axonal degeneration. So the first method we used was transient lipid-mediated siRNA transfection. So this process is introducing silencing RNA, or siRNA, into a cell, which enables us to study the expression of a gene or protein. The siRNA silences the target mRNA to prevent proper translation into proteins. So it's transient because the siRNA only remains in the cell for a short period of time, and we did this for 48 to 72 hours, and lipid-mediated as it uses lipids to cross the cell membrane. So the basic method is taking double-stranded RNA, which has a net negative charge, and cleaving it with a dicer enzyme to form the siRNA. This negatively charged siRNA is engulfed by positively charged lipids to form a transfection complex. This complex enters the negatively charged cell to form RNA-inducing silencing complexes, or RISCs. So this siRNA looks for complementary mRNA to bind to and cleave, and then this foreign mRNA is recognized by the cell as abnormal and then is degraded, and no translation occurs, and therefore the gene is silenced. we used 50B11 immortalized rat DRG, or dorsal root ganglion, neuronal cells for this method. So the second method we used was SDS-Page Western blot, and this is commonly used to detect protein expression. So first off, proteins are denatured by SDS, which coats the proteins in a negative charge, and beta-mercaptoethanol, which breaks the disulfide bonds between the proteins. And this results in proteins separated solely on molecular weight. The protein lysates are then run through a polyacrylamide gel to be separated solely based on molecular weight. And the smaller proteins travel further and faster. The proteins are then transferred to a membrane to be probed by primary and secondary antibodies. In our case, enhanced chemiluminescence, or ECL reagent, and chemiluminescent detection imagers were used to detect the amount of protein that we had. And so light is produced as a byproduct of the ECL substrate and catalyzed by the enzyme found in the secondary antibody. And we used Western blotting after transfection and drug treatment. So lastly, we used an NAD assay to detect the amount of cell death after treatment with chemotherapy. So the basic process is that NAD cycling enzyme converts NAD into NADH. And in the presence of NADH, reductase enzymatically reduces a proluciferin into a luciferin. Luciferin is then detected after addition of a light-emitting luciferase reagent and imaged. The optical density, or the amount of light produced, is proportional to the amount of NAD in the sample. So our preliminary results show that after 48 and 72 hours of transfection, MAP4K4, or HGK, was indeed silenced by siHGK. So you can see this by the control and scrambled siRNA, which the control was nothing and scrambled was just random siRNA, have no difference in the amount of expression of MAP4K4, while the siHGK almost completely eliminated the MAP4K4 expression. And we also included the GAPDH loading control to make sure that we loaded the same amount of protein in each lane. So these are our results of the NAD assay after transfection and drug treatment. So we used PTX, which is a microtubule targeting agent chemotherapy, or MTA, and combined that with a transfection of HGK, TNIC, and MINC, our three proteins that we were interested in looking at. So the NAD levels are proportional to the optical density, and we saw that with the vehicle or the control, the optical density and the NAD levels didn't change with the different transfections. But then we saw that with the chemotherapy, the NAD levels did drop for both the controls and actually recovered with the use of siHGK, si-TNIC, and si-MINC. And so this shows that those three are protective against the PTX drug treatment. So one of the limitations we faced on this project was having to troubleshoot Western blot procedure and antibodies for various proteins. So each protein has its own specific primary and secondary antibody concentration that had to be used to avoid under or overexpression of the protein. And so in the future, we would use siRNA agents in pairing with a different chemotherapy other than PTX, such as cisplatin, and analyze the effects on downstream proteins and apoptosis markers. Also, we would have to repeat the transfection with the other kinases in the MAPK pathway to ensure that the transfection worked properly. Lastly, we could also introduce an in vivo part of this experiment. Lastly, I just want to say thank you to everybody who helped me this summer. Thank you to Dr. Hoke, who provided this wonderful opportunity and lab to do this research. Thank you to Dr. Isel Fiskin, who was a wonderful mentor for me this summer. Thank you to Christina Nance and Catherine Wilson, directors of the SIP and CSM-SIP for Johns Hopkins. Thank you.